Hello and welcome to this Introduction to Microbiology video. I'm Dr. Ali Roberts, and in this video we're going to look at the ways in which we visualize microorganisms using different forms of microscope. Now, unsurprisingly, microorganisms are everywhere. However, whilst our eyes are very good and can see an awful lot of things, they cannot see anything smaller than around 100 micrometers or 0.1 millimeters. And this is a problem for microbiologists because if we want to observe microorganisms, we need to use tools that allow us to see beyond the limits of human vision. Now, in a sort of indirect way, we can see them in our day-to-day life. We can see mold growing on bread, for instance, or in a more scientific capacity, we can see bacteria growing on agar plates. So here we can see Pseudomonas aeruginosa growing on sheep blood agar. Now, whilst we can see this bacteria growing, we can't see the individual cells. Just like if you were to look at a friend, you could see them, but not the individual cells that make up that person. And so despite seeing this collective group of cells, I can't tell you the shape of these bacterial cells, I can't tell you their definitive size, I can't determine whether these cells are motile or unable to move, and I don't know if they have any specialized structures. And this is where the field of microscopy helps. It allows us to magnify microorganisms to the point where we can start to resolve some of these features. And so here we can see Pseudomonas aeruginosa cells that have been visualized using high-powered scanning electron microscopy. And from this we can start to see that Pseudomonas aeruginosa cells are rod or bacillus shaped cells. They measure about 1 micrometer in diameter and anywhere between 2 and 5 micrometers in length. Unfortunately, using this particular method, we can't determine if the bacteria is motile. However, we can start to see some structures. And so if we were to zoom in, we can see some of the bacterial cells are undergoing cell division, and we have the formation of a septa between the two cells as one mother cell becomes two daughter cells. Now, microorganisms come in a range of different sizes. Prokaryotic microorganisms such as bacteria and archaea are typically smaller, ranging from about 400 nanometers to 2 centimeters, and despite their small size, there's huge variety amongst them. For example, Staphylococcus aureus is about 1 micrometer in diameter, whilst Escherichia coli is 2 micrometers, making it twice as large. Then we have Bacillus megaterium at around 4 micrometers, which is twice as big again, with the biggest being Thiomargarita magnifica coming in at around 2 centimeters in diameter for a single-celled microorganism. Now, for the most part, however, we are dependent upon the light microscope to see these cells. Now, in contrast to this, eukaryotic microorganisms, including things like protozoa, algae and fungi, tend to be a little bit larger, ranging from 800 nanometers to 4 centimeters, and again there's considerable variation. For comparison, a red blood cell is around 8 micrometers in diameter, which is 4 times the size of an E. coli cell, while some other cells such as the human ovum at 0.1 mm or paramecium species cells at 0.15 mm are just large enough to be seen without magnification. Now, in addition to coming in at a range of different sizes, microorganisms come in a range of different shapes or morphologies. And several major morphologies have been identified, as we can see here. Now, these are more descriptive names which have more formal designations. Now, one of the most common shapes is the spherical shape known as cocci, and a well-known example is Staphylococcus aureus, a bacterium that can cause skin infections, pneumonia and sepsis. Now, cocci don't always exist alone. They can form different groupings depending on how they divide, and so diplococci form pairs of cells, streptococci arrange themselves into chains, staphylococci grow in grape-like clusters, which is a key characteristic of Staphylococcus species. Tetradcoccus refers to 4 cells arranged together in a square shape, and Sarcina consists of 8 or more cells arranged into a cuboidal structure. Now, moving on, another common bacterial shape is the rod-shaped form known as bacilli, A single rod-shaped cell is called a bacillus, and a well-known example is Escherichia coli, which can be found within the human gut. However, some strains of E. coli can be pathogenic, causing things like food poisoning and urinary tract infections. Now, one particularly dangerous strain of E. coli is known as E. coli 0157H7 and is associated with severe foodborne illness. Now, just like cocci, bacilli can also form different arrangements, which helps in their identification under the microscope. Okay, so from this, some bacteria don't neatly fit into the categories of cocci or bacilli. Instead, they take on an intermediate shape known as a coccobacilli. Essentially, they are short or slightly oval rods that appear somewhat rounded, and a key example of this is Haemophilus influenzae, a bacterium that can cause respiratory infections, including things like pneumonia, sinusitis, and bronchitis. Now, because of their kind of in-between shape, coccobacilli can sometimes be difficult to classify under a microscope, making biochemical tests and genetic identification crucial for accurate diagnosis. Some other bacteria take on a distinctive comma or curved shape known as vibrio. Now, this curvature is due to asymmetric cell wall growth, where one side elongates slightly more than the other during division, and a key example of this is Vibrio cholerae, which is responsible for cholera and severe diarrheal diseases. Now, moving on, we have some of the more complex shapes. Some bacteria have a distinctive spiral shape known as spirilla, and these are essentially rod-shaped bacteria with multiple bends, giving them a rigid helical structure, and it is thought that this shape helps them navigate through their environment. Now, a well-known example is Helicobacter pylori, a bacterium that has evolved to survive the extreme low pH of gastric acid within the human stomach. Now, Helicobacter pylori is closely associated with things like peptic ulcers and has been linked to an increased risk of gastric cancer, making it an important pathogen in gastrointestinal diseases. Some of the bacteria have an even more extreme spiral shape, which forms a corkscrew, and these are known as our spirochetes. Now, spirochetes are highly flexible, allowing them to coil up like a spring, and this shape is particularly useful for burrowing through viscous environments such as mucus or tissue. And a key example is Treponema pallidum, the bacterium responsible for the sexually transmitted infection syphilis. And finally, not all bacteria have a fixed shape. Some are what we call pleomorphic, meaning their shape can vary depending on their environment and growth conditions. Essentially, they don't have a defined structure, and this is usually due to the absence of a rigid cell wall, which can make it very difficult to classify under the microscope, and again requires specialized staining or molecular techniques for identification. And I guess a key example of this would be Mycoplasma pneumoniae, a bacterium responsible for atypical pneumonia and other respiratory tract infections, which has a thick layer of mycolic acid in its cell wall, which contributes to its unusual shape. Okay, now we've learned a lot about microorganisms, including their size and shape, through the use of microscopes, which have become an essential tool in microbiology. They allow us to visualize cells that are too small to see with the naked eye, as well as observe subcellular structures, revealing internal details that help us understand microbial function. Now, the level of detail we can observe depends on the microscope we use, and ultimately the size of the structures we want to see. And so with the human eye, we can only resolve objects larger than around 100 micrometers, about the size of a human hair. To see smaller details, we need microscopes. Now, a compound microscope allows us to see objects down to around 2 to 400 nanometers, making it ideal for viewing cells and larger organelles. Electron microscopes take us even further, letting us visualize subcellular structures, viruses, and even molecular complexes, whereas atomic force microscopy can reveal individual molecules and even atomic scale details. Now, one of the interesting things on this scale is that around 400 nanometers we hit a fundamental limit for light microscopy. Above this, we're generally looking at whole cells known as the cellular range. Below this, we move into the subcellular range, where we can explore the internal components of cells in much finer detail. Now, there are two important concepts to help us detect objects in microscopy. The first is magnification, which refers to the increase in the apparent size of an image. It makes objects look bigger, but doesn't necessarily add more detail. And the second is resolution, which is the ability to distinguish fine detail, specifically the ability to distinguish two close objects as distinct objects rather than a single blurred shape. Essentially, magnification enlarges the image, whilst resolution ensures the image remains clear and detailed. Now, one of the biggest factors affecting resolution is actually the wavelength of light. And as you might know, light is part of the electromagnetic radiation spectrum, and therefore light is a wave associated with both electrical and magnetic fields. And so if we were to look at a photon of light, we can see there is an electrical field and perpendicular magnetic field. Now these fields move in waves, and the distance between each is known as its wavelength, which different lights have different wavelengths. So the wavelength for visible light, which helps us to see, is somewhere between 400 and 750 nanometers, with red light having larger wavelengths that decrease as we go through orange, yellow, green, blue, indigo, before we reach violet light, which has a shorter wavelength around 400. Now I know what you might be thinking, why is this important? Well, the wavelength of light and the properties of an object affect how the two interact, with the photon of light being altered in one of four ways by an object. And so a photon of light can be absorbed by the object, it can be reflected away from it, it can be refracted through the object, or it can be scattered away. And we will have a look at each of these in a little more detail, starting with absorption. This occurs when an object takes in a photon's energy, typically converting it to heat. Now in conventional microscopy, absorbed light renders microscopy specimens as dark areas against a bright background. Next is reflection. This occurs when a photon of light hits a surface at an angle, known as the incident angle, shown by the theta symbol, and is redirected at an equal angle, known as the angle of reflection, shown by the theta prime symbol. Now this principle is essential in microscopy optics, where mirrors and glass surfaces within the microscope guide light paths to form images. Next we have refraction. This is where light goes through an object and slows down, causing it to bend slightly. Now the extent of bending depends on the refractive index of the object, which is a measure of how much an object can alter the speed of a photon compared to air. And again, this principle is essential in microscopy optics, as it enables lenses within the microscope to magnify images. More on this in a minute. And finally, we have light scattering, which occurs when light interacts with an object and a portion of the light is redirected in all directions, forming a spherical wave. Now, the issue with this is if we have multiple objects that are close together, with each scattering light in all directions, then scattered light might overlap, creating a visible haze that obstructs fine detail, which is problematic for light microscopy. Now when it comes to magnification within a microscope, we need to bend light, which if we remember is known as refraction. But the key question is, how do we accomplish magnification through the process of refraction? Well, when light moves from one medium, such as air, into another with a different refractive index, such as glass, it changes speed and the light waves bend. Microscopes then exploit this, directing the bent light in a controlled manner to produce a magnified image. And so for microscopes to produce a magnified image through refraction, they rely on parabolic glass lenses. These lenses have a curved shape and a refractive index of around 1.5, which means they slow and bend the light as it passes through it. Now, the refractive index is a measure of how much light changes direction when moving between two different materials, and so the higher the index, the slower the light moves and the more the light will bend. Now, when light enters a parabolic glass lens, it is refracted, causing the light waves to converge on a central focal point. The distance to this point is known as the focal distance, which depends on things like the lens curvature and its refractive index. However, by carefully designing these properties, microscopes can focus light precisely, producing clear, magnified images of microscopic specimens. Now, if we were to look at the light converging at the focal point, we know it originates from an object with a real physical size. However, if our eyes were positioned at this focal point, we wouldn't perceive the bending of light. We would see the light as if it were travelling straight towards us, as if it hadn't been refracted at all. This means the object appears much larger than it actually is. And so by manipulating how light converges and is redirected through lenses, microscopes take advantage of this effect to create magnified images with an increased apparent size, allowing us to see microscopic details that would otherwise be invisible to the naked eye. Now, unfortunately, when a microscope increases the apparent size of an image without improving the level of detail, we call this empty magnification, and it is commonly associated with a blurry image. And so if we want to make full use of a magnified image, we also need to increase its resolution, which, if we remember, is the ability to distinguish detail between two distinct objects. And sadly, it is resolution that is a key limiting factor when it comes to light microscopy. Now, the limit of resolution for a light microscope is around 0.2 micrometers or 200 nanometers. And what this means is that if two objects are closer together than this, they cannot be distinguished as separate entities. Instead, they will just appear as a single blurred object, and this limitation is fundamentally linked to the wavelength of visible light. Okay, so we're now going to move on and look at several microscopy techniques. But let's start by talking about the compound microscope. This instrument relies on multiple lenses working together to minimize distortions known as aberrations that occur when light bends through curved glass. But why not just use a single high-powered lens? Well, the problem is that as magnification increases, these distortions become more pronounced, making it difficult to produce a clear image. Instead, the compound microscope uses a series of low-powered lenses that combine their magnifying power while keeping aberrations to a minimum. And so if we take a closer look at the compound microscope, at the base we have the light source, which produces the illumination needed to view the specimen. The condenser lens focuses the light onto the sample, optimizing illumination and contrast for clearer imaging. Next, the objective lens collects light from the specimen, providing the primary magnification and resolution, as this is where the fine details of the sample are captured. The ocular or eyepiece lens provides an additional magnification step and helps reduce that overall optical aberration. Now, typically, most compound microscopes will come with multiple lenses to increase magnification, and typically within microbiology, we would pair a 10 times ocular lens with a 100 times objective lens to achieve a total magnification of up to 1000 times. Now, a good thing about compound microscopes is that they are typically parafocal, meaning they maintain focus when switching between different objective lenses. This allows for a smooth transition between magnifications, requiring only minor, fine adjustments rather than refocusing from scratch. And a good way to achieve the best focus is to start at a lower magnification, Around 40 times or 10 times, since it's easier to locate and focus on the specimen, and then once a specimen is in focus, we can work up to the higher magnifications such as the 100 times for greater detail, often using oil immersion to enhance clarity and resolution. But what is this oil immersion? Well, this technique involves placing a drop of oil between the sample and the 100 times oil immersion objective lens. The oil allows the lens to get closer to the specimen whilst minimizing the loss of light and limiting distortion. Now, for oil immersion to be effective, the oil must have a refractive index that matches both the glass slide and the glass lens. And what this will do is it will minimize refraction and maximize light transmission, ensuring that more light enters the objective lens. And the result will be a clearer, sharper image with improved resolution at higher magnifications. Now that might not have made much sense, so let's have a look at this visually. First, we place the specimen in the center of a glass slide and view it under the 100 times objective lens for high magnification. Now without oil, when light passes from the glass slide into the air before reaching the lens, it refracts away, causing significant light loss and reducing the image clarity due to the large air gap. If we add a drop of immersion oil between the sample and the lens, and it has the same refractive index as the slide and the lens, we will create a continuous optical path. This allows more light to be captured by the objective lens and minimizes refraction, all whilst preventing light loss, resulting in a much sharper and clearer image. Okay, so finally, we're going to look at some different microscopy techniques, starting with the most common technique known as bright field microscopy, which is where an object appears silhouetted against a bright background. Essentially, an image is generated due to differences in light absorption, refraction, and scattering as light passes through an object. The object will absorb or scatter light, appearing darker, whilst transparent regions remain brighter. Now, there is a problem with this in that many biological cell samples we might want to look at down the microscope are transparent. Therefore, we often use stains to try and enhance contrast between the cell and the background. So here we can see a bright field image of some tissue fibers. The dark fibers in this image are absorbing light, preventing it from passing through, which makes them appear darker against the background, whilst the bright background results from unobstructed light passing through the slide, creating contrast between the sample and its surroundings. Next, we have dark field microscopy, which is a specialized light microscopy technique that enhances contrast by illuminating the sample with oblique light, essentially light that has been scattered from the sides, rather than direct light passing through. Now, this method is particularly useful for observing thin, transparent or unstained samples that would otherwise be difficult to see under bright field microscopy. Since dark field microscopy relies on light scattering, it can reveal fine details such as the edges of delicate fibers, small bacterial cells, or intracellular structures without the need for staining. And so here we can see a dark field image of some tissue fibers. The tissue fibers appear bright because they scatter light in different directions, making them stand out against the dark background, which has direct light blocked by special filters. And finally, we have phase contrast microscopy. This technique exploits differences in refractive index between the cytoplasm and its surrounding medium, as well as between different organelles within the cell. And so by converting these variations into differences in light intensity, phase contrast microscopy enhances contrast without the need for stains or dyes. This makes it particularly useful for viewing live cells, allowing us to observe internal structures and cellular organelles in their natural state. And so here we can see a phase contrast image again of tissue fibers. In this image, the fibers appear different because they have varying refractive indexes, which the microscope translates into differences in light intensity. This highlights structural details that would be difficult to see with conventional bright field microscopy, and again, this is all without the need for staining. Now moving on, but still using light to visualize cells, we have fluorescent microscopy, and this allows us to visualize specimens labeled with fluorescent dyes or proteins under specific wavelengths of light. And so what we call a fluorophore is added to the sample, binding to specific structures within the cell. And when this is exposed to light of a very specific wavelength, the fluorophore's electrons absorb this energy and move to a higher energy state. As they return to a lower energy state, they emit light at a longer wavelength, producing fluorescence we see in an image. Now, this technique provides high specificity, allowing us to highlight individual cellular components with exceptional clarity. So here we can see a fluorescent microscopy image of some bacteria. And in this image, different Escherichia coli cells are expressing one of 6 different fluorophores, so that they appear as either cyan, magenta, yellow, red, green, or blue. This can help us to look at how bacteria interact and associate with one another in surface-associated communities. And we now move on to our final microscopes, which are the electron microscopes. Now, unlike the light microscopes, electron microscopy uses beams of electrons which also move in waves but have a much shorter wavelength, at around 100,000 times shorter than visible light. This dramatically increases both magnification and resolution, allowing us to visualize structures at the nanoscale rather than the microscale. And so for our electron microscopes to work, samples must be able to absorb electrons, so they are coated in very heavy metals such as gold. The sample is then placed in a vacuum where electrons are fired at the sample, and instead of glass lenses, electromagnetic fields help to focus the electron beam, creating highly detailed images of cellular structures and even molecular complexes that are invisible under traditional light microscopy. And I guess finally, here we have a scanning electron micrograph, or SEM, showing bacteria amongst red blood cells in the circulatory system. The SEM image provides a detailed 3-dimensional view of the surface structures by bouncing electrons off the sample, allowing us to see various bacilli-shaped bacterial cells covering erythrocytes. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.